Skeletal muscles are made up of fascicles, which are bundles of muscle fibers encircled by connective tissue, blood vessels, and nerves. Skeletal muscles are voluntary and regulated consciously by the neurological system. A motor unit is made up of one or more skeletal muscle fibers that are supplied by motor nerve axons. Myofibrils, which resemble thread-like structures, are arranged inside muscle fibers into contractile units, or sarcomeres, which mimic the striations present in skeletal muscle. Myofilament strands of the proteins myosin and actin, both thick and thin, make up each sarcomere. Myosin creates dense filaments. Actin builds up into tiny filaments. Myosin filaments with projecting heads create cross bridges with actin filaments and the energy transfer molecule ATP to bind to them. A series of actin molecules with myosin binding sites can be found in thin filaments. Actin binds tropomyosin and troponin to contractile proteins that prevent contraction when the body is at rest. Transverse or T-tubules that surround each myofibril and are open to the extracellular environment are the first invaginations to form during the contraction of skeletal muscle at the plasma membrane, or sarcolemma, of the muscle fibers. The sarcoplasmic reticulum, a membranous network connected to T-tubules, regulates muscle contraction by storing calcium and transferring ions into the myofibril sarcoplasm through voltage-gated channels. A motor nerve fires an impulse or action potential during the first phase of muscle contraction, known as excitation. Action potential that travels across the sarcolemma and into the T-tubules, depolarizing the junction, depolarizing the sarcolemma, and creating an action potential that stimulates the muscle fiber. The action potential depolarizes the T-tubules in the second phase, coupling. Calcium is released into the sarcoplasm through channels in the sarcoplasmic reticulum. Troponin binds calcium and separates from tropomyosin along the myofilaments. The actin-binding sites on tropomyosin are exposed in the third step, contraction, allowing myosin to bind to actin and activate ATP and cross bridges. The myosin heads rotate as a result of the energy released, pulling the thin filaments inward where they slide past the thick filaments and shorten the sarcomere. The myosin-actin connections are broken during the fourth stage of muscular contraction, relaxation, in which calcium ions return to the sarcoplasmic reticulum. The muscle relaxes and the contraction comes to an end as a result of the cross bridges disengaging and the sarcomere lengthening.